I'm Daniel Levine, and this is the Bio Report. About 98% of the human genome consists of non-protein coding regions known as the dark genome. Once derided as junk DNA, these regions are increasingly being understood to play a critical role in the regulation of the genome and offer a novel means of targeting diseases. Haya Therapeutics is exploring long non-coding RNAs as potential therapies to treat a range of diseases. Haya Therapeutics is exploring long non-coding RNAs as potential therapies to treat a range of diseases. We spoke to Samir Ounzain, co-founder and CEO of Haya, about the dark genome, the potential to use lncRNAs to treat diseases, and its recently announced collaboration with Eli Lilly to use Haya's platform technology to discover therapies for obesity and related metabolic conditions. Samir, thanks for joining us.
Thanks so much for the invitation, Danny. I'm really happy to be on this podcast.
We're going to talk about Haya, the dark matter of the genome, and how Haya is developing therapies to target both rare and common conditions in which the non-coding portion of the genome is implicated. Let's start with this non-coding portion. This has been referred to as junk DNA. How well do we understand the non-coding portion of the genome, and how is our understanding of this emerging and changing?
Absolutely. So yes, the dark genome, as some people call it, this really represents the 98% of your DNA that doesn't code for proteins. So obviously throughout the history of molecular biology and drug development, we've really had a focus as proteins is the main protagonist in molecular biology and ultimately disease-driving processes. And since the human genome was sequenced over 20 years ago now, we had this very big surprise that actually most of your human genome doesn't code for protein. And at the time, it was, I think, somewhat prematurely referred to as junk DNA, was thought to really be an accumulation of parasitic, non-functional junk that's accumulated over evolutionary time. But I think what's happened over the last 20 years in particular with the accumulation and the integration of all these fantastic technologies, sequencing-based technologies and data science capabilities around them, is we've actually started to understand that the non-coding genome or the junk DNA, it's actually the information processing portion of the genome. You can think of it as the software of the genome. It's the part of the genome that actually directs and coordinates the activity of the building blocks of your cells, the proteins, telling them where to go at what time in what context. And I think what's really rapidly emerging, even in the more recent past, is the different mechanisms that are involved in the activity of the regulatory genome or the dark genome. So you can really think of this as where all of the switches are, the so-called regulatory elements like enhancer elements that can coordinate and allow the genome to speak with your environment and direct the activity of pathways which ultimately control cell identity and cell state. Now, I think why this is becoming a very interesting area is, especially when we start to look at common and chronic diseases, we've learned a couple of things over the last 10, 20 years, is that most of these common and chronic diseases, the functional unit that causes the disease is changes in cellular behavior or cellular state. And how those cells change state is how they respond to the environment. And so you have this process of epigenetics, which is how does the cell respond to its environment, which is actually hardwired in the junk DNA or the dark genome. And you also have the emergence of all of these genetic studies for common and chronic diseases, which have actually mapped all of the genetic variation that predisposes you and creates a risk for developing many of these diseases that plague society. And we had another big surprise, which is over 90% of all of this genetic variation associated with these diseases is also in the junk DNA. So I think we have two things, which is the epigenetics that controls how cells respond to the environment, and the genetic variation that predisposes you from a risk perspective to developing these diseases is hardwired in the dark genome. And I think that's where we are now in terms of the understanding. And finally, I just say the biochemical and the molecular processes. So we're understanding how different converging biochemical processes, for example, the formation of condensates, which is an emerging area of also therapeutic interest, is central to the activity of the dark genome. It allows the dark genome to form three-dimensional structures to respond to the environment. And last but not least, which is where really Haya is playing in this space, is the emergence of RNA as the substrate that really orchestrates and coordinates the activity of these mechanisms and how the dark genome responds to the environment to control cell state, which ultimately is the fundamental unit of disease-driving processes in many of these diseases.
This is an area that you focused on as a postdoctoral researcher. How did your work then lead to the formation of Haya?
Yeah, so I'd spent my academic career really focusing on how does the dark genome control, in particular, cardiovascular development in disease. So the work I was involved in was some of the first work that really characterized and built atlases of the dark genome in cardiovascular development. So how are the cells, for example, in the heart specified during development to control the formation of the heart tissue and the different cell types within the heart. But then the work also transitioned to look, how does the heart respond to stress and different disease-driving processes associated with diseases like heart failure? And what is the implication of the dark genome in those processes? And that academic work led to the discovery. We actually discovered many of the first and best characterized novel RNAs derived from the dark genome. The technical term being, for your audience, long non-coding RNAs, or lncRNAs for short. We mapped and discovered these molecules in the context of heart disease. And in particular, there was one process where we really were interested in these targets, which was in fibrosis within the heart, which is a really central disease-driving process that underpins one of the fundamental domains of myocardial vulnerability and remodeling that leads to heart failure, which is this process of fibrosis. And what we actually discovered were a number of these RNAs that specifically only in the heart control the activity of the effector cells that cause fibrosis, which are called myofibroblasts, which is a disease-driving form of a activated fibroblast. And that was really the discovery that led to the transition to the founding of Haya. Because what has been apparent in the fibrosis space, and I'm sure as your audience is aware, 1 in 3 people actually at some point will succumb to illnesses caused by fibrosis. Yet we have very limited targeted and specific anti-fibrotic therapeutic opportunities. And one of the reasons for, I would say, the lack of progress in the fibrosis space is historically we've been focusing on the proteins that control fibrosis. So think of pathways like TGF-beta signaling. Now, these protein-based pathways that control fibrosis are very pleiotropic. And this is because proteins themselves are not very specific. The same proteins and pathways can be used for different purposes in all of the different tissues and organs and cell types within your body, which means when you're trying to develop drugs against these targets, these proteins and pathways, typically what happens is we've seen historically in the fibrosis space is by the time you get to a phase 2 clinical trial, you have a very limited therapeutic window due to exaggerated pharmacology outside of the fibrotic tissue you care about, which limits efficacy and typically leads, unfortunately, to clinical failure. And what our work has shown is through the dark genome and through these RNAs, we can start to identify very important regulators of fibrosis or fibroblast activation, but specific to the tissue and the disease context. So you can now start to imagine a precision tissue-specific approach to antifibrotic drug development. And that was really the genesis, the concept that led to the founding of Haya Therapeutics.
What's understood about the way the long non-coding RNA regulate genes and pathways?
When I talk about these long non-coding RNAs, I like to think of them as very sophisticated Swiss Army knives, because what they can actually do is they can confer on intrinsically, I would describe them as analog or non-sequence selective proteins, sequence-based specificity. So they can confer specificity at the nucleic acid level onto proteins. So this, you can imagine, becomes very important when you're thinking about epigenetics. Most of the proteins that lay down epigenetic marks themselves don't have intrinsic specificity to target specific regions of the genome. These long non-coding RNAs can offer that specificity. So think of them as specialization factors conferring sequence and context-based specificity on intrinsically analog proteins that themselves are not very specific. And maybe last but not least, which I think is really an emerging area of biology, where there's also a lot of therapeutic interest, is this concept that there are many different partitions, membraneless partition domains within the cell that concentrate biochemical activity that is very important for protein functions and pathways, the so-called membraneless condensates, which are these condensates that concentrate biochemical activity and allow proteins to do their thing. And I think what's really rapidly emerging, actually, over the last number of years is the biomolecule that is really important in controlling both the genesis and the dynamics of these condensates. It's actually these RNAs, these long non-coding RNAs. So by conferring sequence-based specificity and scaffolding and seeding functions for condensate formation, they're really emerging as very potent pharmacodynamic regulators of disease-driving cell state processes. And they're extremely specific in terms of their expression and activity, which renders them quite attractive from a targeting perspective.
And would you be able to just turn these on or off, or are you able to actually modulate the level of activity they have?
So using the emerging genetic medicine toolbox of modalities, you can actually start to drug them in slightly different ways. So you could drug them in a way where you literally degrade them. So you can imagine here using modalities like antisense oligonucleotides or small interfering RNAs. But you can also drug them in a different way, which is instead of degrading them, you can interact or block how they interact with the proteins, which is what they coordinate from an activity perspective. Also using oligonucleotide-based therapies, which could, for example, mask binding sites within these RNAs to influence their ability to scaffold and interact with proteins. And there's also efforts now that are developing using small molecules to interfere with the ability of these RNAs to interact with other biological molecules, in particular proteins. But the other thing I should mention around the kinetics and the activity of these RNAs, they're very rapid in responding to environmental signals. So it requires transcription, which can be a very, very fast process, leading to a very rapid biochemical activity through these phase separation mechanisms, which also, depending on the durability and the half-life of your targeting modalities, can give you different ways to modulate those responses. So there's a lot of programmability around this target class that I think can be leveraged depending on the target, the indication, and the disease-modifying effect you're trying to elicit.
Well, let's talk about your lead experimental therapy. This is a treatment for hypertrophic cardiomyopathy. Can you explain what this condition is?
Sure. So hypertrophic cardiomyopathy is a relatively rare structural heart disease, which is associated with the thickening of the heart. So you have hypertrophic thickening of the heart that impacts the structure of the heart, which is significantly associated with fibrosis, that unfortunately ultimately leads to some very undesirable outcomes, specifically heart failure. Now, one of the interesting things about hypertrophic cardiomyopathy is that one of the major drivers of symptoms and ultimately outcomes in these patients who are suffering with this. So just in terms of the patient population, around 1 in 500 people will unfortunately suffer from HCM. There's around 700,000 patients diagnosed today in the United States. And of that group of patients, there are in particular around 300,000 who suffer from a form of this disease that is primarily associated with cardiac fibrosis and diastolic dysfunction. Now, unfortunately, symptoms can progress over time, and ultimately they can go on to develop heart failure and be at very high risk for example, sudden cardiac death. So these patients really represent a very significant unmet need, in particular for disease-modifying therapies in the heart that can really affect the remodeling and the structural disease-driving processes that cause these outcomes.
How difficult a condition is this to diagnose, and how dependent is the outcome on how early the diagnosis is?
Yeah, so diagnosing hypertrophic cardiomyopathy can be challenging because, first of all, very often the symptoms like shortness of breath or chest pain can often mimic those of other types of conditions, like, for example, hypertension and valvular disease. And also, I think it's important to mention that a lot of patients who are carrying the genetic mutations that can predispose you to HCM can very often be symptomatic or have very mild symptoms for quite a long period of time. And typically, many individuals could be diagnosed only after a cardiac event or having a family history. So HCM is very often a genetically associated disorder. And of course, a family history can give you an insight into your potential risk for developing this disease. So typically patients are found once they're symptomatic, they have these symptoms of heart failure. And typically at that point, actually, fibrosis is the major driver of the symptoms and the outcomes, which I think again illuminates or highlights why antifibrotics for this patient population could be highly relevant.
And how is the condition generally treated today? Are there existing therapies?
So we've had some really exciting progress over the last 2 to 5 years, which I can touch on. I think these are new medicines that have been approved, the so-called cardiac myosin inhibitors. What they do is they reduce the hypercontractility in the heart that can lead to some of these symptoms as a consequence of the genetic lesions, which typically you find in the sarcomere and the myosin proteins. So this is really good news for patients with obstructive hypertrophic cardiomyopathy, because these patients typically were relying ultimately when they had severe symptoms on surgical interventions, for example, septomyectomy. And unfortunately, many of these patients would ultimately need to have an ICD or ultimately heart transplantation. So there is emergence of new therapies, the so-called cardiac myosin inhibitors, which are making an impact and I think are bringing a lot of hope. But there's still a lot of issues around how do we treat these patients, in particular, the 30% of HCM patients who do not have obstruction. So by obstruction, what I'm referring to is with this hypercontractility, this can lead to obstruction of the outflow tract. That is an early driver of early symptoms, which can be treated to some extent with surgical or cardiac myosin inhibition. However, the non-obstructive patient population, which represents about 200,000 patients, they become symptomatic really as a consequence of diastolic dysfunction when they have heart failure. And these patients, sadly, today have essentially very, very limited options. Ultimately, the ultimate option for these patients as they progress through developing heart failure ultimately is heart transplantation. So this subset of patients really need disease-modifying therapies that target the remodeling in the heart that leads to diastolic dysfunction. And from this perspective, this is the problem we're trying to solve. Let's bring these patients an antifibrotic approach that can both block the remodeling and ultimately potentially reverse remodel the heart to reduce their probability of these really devastating outcomes that they suffer with.
Well, what is your experimental therapy and how does it work?
Sure. So our experimental therapy is a antisense, so it's a synthetic modified antisense oligonucleotide that, when administered, is able to engage this RNA that we've discovered that is specific to activated cardiac myofibroblasts within the heart. So this is a long non-coding RNA we discovered called WhisPR, which we've demonstrated across a number of different animal models, as well as validated the target and the pathway as being associated with fibrosis in many different heart failure associated indications, is really emerging as a master regulator that drives the activation and controls the cell state changes associated with fibrosis. So it controls fibroblast programming to an activated state. Now, when we engage this target using an antisense oligonucleotide, what we're essentially able to do is reprogram the activated myofibroblasts in the heart back towards a more healthy quiescent state that blocks the progression of fibrosis. Furthermore, we also, through this mechanism of action, have indications that we can not only block fibrosis progression, but potentially we may have the ability to also reverse remodel the heart by reversing established chronic interstitial fibrosis, which ultimately would really be a game changer for patients suffering with chronic heart failure. Both patients suffering with non-obstructive hypertrophic cardiomyopathy associated diastolic dysfunction, but ultimately more larger heart failure patient populations. For example, those patients who are suffering more broadly with heart failure with preserved ejection fraction, which represents all of heart failure and actually will ultimately impacts around 1 in 10 people.
And what's known about it from studies you've done to date?
So what we've been able to demonstrate in both rodent and large animal preclinical models is that these antisense oligonucleotides, when administered, are able to reprogram these cells in vivo, and that in vivo reprogramming of this myofibroblast activation is able to significantly reduce the expansion of the interstitium associated with fibrosis. So when I say the expansion of the interstitium, I'm really referring to the deposition of collagens and extracellular matrix proteins that these myofibroblasts, the effector cells of the disease, deposit in the myocardium, which results in impaired compliance, myocardial compliance. It increases the stiffness of the heart. And this is really the process actually that results in diastolic dysfunction and heart failure. So by targeting this long non-coding RNA, we can both block and we also see evidence of reverse that pro-fibrotic signal.
And is the expectation this would slow or halt progression of the disease, or is there a possibility you could actually reverse it?
So the minimum expectation is to block the progression of the disease, but some of the early data and signals we're seeing in some of our preclinical animal models also give us excitement that we could also potentially achieve a reversal in different contexts, depending on the indication of the patient population. And that, of course, is really an exciting opportunity when you think about heart failure more broadly. And there is a precedent there. I mean, there is actually a way to reverse remodel the heart in humans, which is using, for example, left ventricular assist devices, which in patients with successful LVAD, you do actually see this reverse remodeling. And what we believe is, and what's been demonstrated also, is that a lot of that reverse remodeling is mediated by these cells, the myofibroblasts that are very active and proliferating in the heart. So if you can reprogram them, there is the opportunity, due to the myocardial functional reserve within the heart, to achieve a reverse remodeling effect.
What's the development path forward?
So now with our lead asset, HTX-001, we obviously have nominated a development candidate, which is currently undergoing GLP IND-enabling tox and safety studies. And all being well, we're hopeful that in the not too distant future, 12 to 18-month window, we could be looking at a point where we can start to think about entering into the clinic with this asset. So we're really excited to take this asset through our final GLP tox studies and bring it to the clinic, and hopefully as fast as possible bring this antifibrotic effort to patients.
Haya recently announced a collaboration with Eli Lilly to use its platform technology to discover therapies for obesity and related metabolic conditions. Can you outline that deal for us?
Sure. So the platform around mining the dark genome for these long non-coding RNAs can really be applied beyond fibrosis. So as I mentioned up top, when you think about these common and chronic diseases, including obesity, it's really a response of cells to the environment. And what we can provide, and the objective of this exciting collaboration with Eli Lilly, is really to provide a perspective on novel RNA targets that could potentially be involved in finding new approaches to combat obesity and related metabolic diseases, where I think there's a huge opportunity for cell state reprogramming. So the collaboration is really leveraging our platform and our expertise in mining and understanding the dark genome. And of course, Lilly bringing an incredible wealth of know-how in how to develop especially genetic medicine approaches going forward in the obesity and metabolic space. So we're excited to get this going.
How validating is this deal for Haya's approach?
So we believe it's extremely validating because it's really demonstrating the power of our platform to deliver disease-modifying cell state reprogramming RNA targets that can have potentially truly breakthrough impact in many common and chronic disease areas. And we think this collaboration with Lilly hopefully really shows the potential of this biological target space. I mean, I should just add, it's obviously very exciting for Haya to see the belief in our platform, but I think it's really exciting for the whole field. So there's many emerging companies that are looking at the dark genome as a next generation biological target space for new medicines. And I think this deal hopefully really catalyzes more broadly the interest in the dark genome as a therapeutic target landscape.
It's interesting because I think of obesity being a fairly complex condition, at least from a genetic level. What does this say about how broadly applicable the technology might be, and how does this inform the way you might think about prioritizing your pipeline?
Yeah, no, it's a really good question. So just up top, our platform has already delivered novel disease-modifying targets for other fibrotic indications or other indications associated with fibroblast cell state transitions. So think of pulmonary fibrosis, in particular associated with IPF, where we have our second program focused. But we've also deployed this platform to look at fibroblasts within the tumor microenvironment of solid tumors, where we know that activated fibroblasts within the TME contribute significantly to outcome and survival in a number of different oncological settings. And there we also have some very exciting data. So broadly in fibrosis, we think the platform has a lot of opportunity. But I think what the collaboration with Lilly potentially highlights is beyond fibrosis. And I think the way I would frame this is, if you think about the spectrum of common and chronic diseases that are associated with common genetic variation, dysregulation of the epigenome, including thinking about diseases associated with aging, and diseases where having a highly context and tissue-specific target linked to the pathogenesis, where cell state reprogramming could bring a lot of value. These are all the type of processes that guide indication selection. And obviously, we're still a relatively young company. We already have a lead program moving towards the clinic, second programs in pulmonary, and we're definitely now at a stage, especially off the back of this Lilly collaboration, where we're trying to assess where brings the most value for our platform in terms of these larger opportunities.
How is the company funded, and how far will existing cash take you?
Yeah, so company, we're a venture-backed company. We raised our seed round just over 3 years ago now. So the company raised a $25 million seed round with a very strong investor syndicate with expertise in different areas, with in particular in the cardiovascular and also in the health span space. And this, coupled with some non-dilutive financing that the company's received over the last couple of years, as well as proceeds from the recent collaboration with Eli Lilly, really puts us in a strong position now to take both our lead program as well as follow-on targets towards the clinic. Now, I think the excitement that we have for our platform to deliver disease-modifying therapies around long non-coding RNAs, as well as our proximity to the clinic, is definitely driving us now to look probably in the mid to near term to further finance the company, to really put us in a very strong position so that over the next 2-3 years, we can achieve first in biology, meaningful clinical proof of concept around this biological target space. Obviously, as we mentioned, Haya is really has a very unique platform behind target identification and candidate development. And we really want to demonstrate the clinical power of these targets in the near term to really, I think, build the value around the platform that is now being built. So we'll be looking to further augment our capabilities to deliver lncRNA targeting medicines to patients as soon as possible.
Samir Ounzain, CEO of Haya Therapeutics. Samir, thanks so much for your time today.
Thank you so much, Danny. Really appreciate the opportunity to come and speak to you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.